Oh, your sound is off? Hello. Yeah. Okay. Hi, Susan. And is this Mayok or Mainak? Okay. Hello, Mainak.
Hello, Bradley. Can you hear me?
Yeah. Yeah.
Okay. Hello. It's morning there, right? So good morning,
Good
everyone.
morning. Yeah. For me and Susan, it's morning. So yeah, I don't know, Susan. If you want to just type into the chat, if you know when you have something to say. I know your audio isn't working. Let's see. Okay. Yeah. Yep. So yeah, Susan's audio is off, and her video's on, but her audio's off, and vice versa apparently. So welcome to the meeting. I don't know who else is going to show up. Usually people kind of filter in, but we have a bunch of stuff to talk about today. Mainak has some updates on the DevoLearn site. Actually, he had some things on the DevoLearn software he was updating. And I know that Mayok has released a new version. I mentioned it in the email. We have 0.2.1. So that's a new version. There were some updates that I think Mainak made, and there were a couple other updates that he wanted to put out a release. And I told him, yeah, if you want to put out a release, we don't really have a formal release schedule, but so we have one. And Mainak will give us an update, and maybe we'll do another release soon.
Yeah. So speaking of my update, like I've got a few slides ready.
Okay.
So whenever you say, I'll just present it. Okay.
All right. Well, let me go through what we're going to do today first.
Yeah. Okay.
So
Okay. Absolutely.
yeah, I don't know if Krishna is going to show up, but he has his evolution abstract. We have the flash talk for an upcoming lecture to the Open Science Framework Conference next week. So it's next Tuesday. I'm going to go through some of those slides, and then we're going to try to work on them this week. And then some other opportunities. Darwin Day's coming up, and then hi Dick, how are you?
Oh, good morning.
Morning. We have and then we have some papers and things like that. So, Mainak, why don't you present your screen?
Can you hear me now?
Oh yeah, I can hear
Yeah.
you.
Can you hear me?
Yeah, I can hear you. I can hear
Yes,
both of you.
sir.
Okay. I'll present my screen. Okay.
Yeah.
I unplug my visual and I get audio.
Oh.
Yeah. Can you
Okay.
see my screen now?
So maybe that'll work.
Okay.
I'm not presenting, so.
Yeah, that's it.
Yes.
Yeah. So can you all see my screen?
Yes.
Okay, so for the past couple weeks, I think more than that. So for the past couple weeks or so, I've been working with the EPIC worm embryo data set. So what happened is that I was going, so first I'll talk a bit about the data set itself first. So this is what the website provides us. We get 260 videos of embryos with separate annotation files for each video. So what Mayuk had done last year is that he had wrote a piece of code which basically translates everything into frames and also creates a single unified annotation file, which makes it easier to train. But the downside was that he was not using all of the 260 videos. He was using 6 or 7, if I remember correctly. But I don't think that was the right thing to do. So what I decided is that I decided to expand on the dataset. So the problem is that we have the current models in the DevoLearn. So what the problem is basically that the current models in the DevoLearn, what we have today, everything was trained on a dataset containing about 3,500 unique data points. But like having such a low amount of data is not really good because it would be difficult for the models to generalize when it has a small amount of training data. Right? So the solution to this problem is that The first solution is to train over more epochs, which could lead to overfitting, which is absolutely something we don't want. And the second option is to use more data, which is just what I did. So Mayuk's dataset has the dataset that Mayuk has scraped contained 3,500 points, but the one that I scraped recently, it actually has many versions. Like I scraped one version that has 10,000 points, and then I also scraped another version which has 25,000 data points, and then I also scraped the largest version which has 50,000 data points in case I need it. So moving on. Okay, so this is basically an example that shows the comparison between using less data and more data. So to the left, we see these blue data points, right? The blue ones are the data points, and the red line is the model prediction. So to the left, this shows a diagram that basically represents what happens if the model is served two less data. Increase the number of data points, we successfully capture the true distribution of the data, which is shown to the right. Like we have considerably increased the number of blue points, which is the data points. Hence, the model is able to more generally classify the objects. So moving on. So we all saw these results, right? So I actually found something really similar going on when I trained it on the lineage population prediction model. So what I basically did is that I trained two neural networks with two different data sets of different sizes with the same hyperparameters like the learning rate, the architecture learning rate, the batch size, etc. So the dotted line here, this line represents the model predictions, and this non-dotted line. So these non-dotted lines represent the targets, which is the actual data that we had. So the architecture which I used in both these cases is the ResNet 50, which in case you were is the ResNet 50. So which is not same of the DevoWorm model. So like this can't be really compared with the results from the current version of DevoWorm, because DevoWorm uses ResNet 18, which is smaller. So anyways, I used the same architecture and the same hyperparameters for both these. So like there's a clear indication that using more data points is helping. Like in the image below, we see that the dotted line is much closer to the blue non-dotted line, which clearly indicates that using more data points is helping. Like it's just like this one, like this image that I showed. It's basically a real life example on the EPIC dataset of in this case. So that's the result I got. Yeah. So as I said before, I have compiled multiple versions of this expanded dataset, and with varying number of data points. Like one I have with 1,000 data points, then 10k, 25k, 50k, and so on. So due to computational limits, it might not be really viable to retrain all the models on the new large data set, which is about 14 times larger on what it was trained on. So in most cases, I guess heavy image augmentation should yield similar results. And plus, like my computer, it's using an MX150 GPU, which is quite slow. It took about 2.5 hours to train the last model that I had shown, this one. So like I might contact Mayuk because he has a better GPU. So I'll just send him the bigger data sets and he'll just train it for me. So that's something that I plan to do soon. And yeah, okay, so that's basically it from my side. And
Okay.
thank you for your time, everyone. Yeah.
Yeah, that's good.
Yeah, thanks for letting me present. Yeah.
Oh, could you share your screen again? I wanted to go back over some of the slides just to talk about them a little bit.
Yeah, of course. I'll do that. Okay.
Any comments on that, or I mean, it looks pretty good.
Okay, can you see my screen?
Yeah, it's coming back up.
So, which slide should I go to?
Well, let's see. Go back. I'll tell you when to stop.
Okay. You can see my screen, right?
Yeah, I can see it. just go back in the slide deck. So this one here, I guess it's slide 4.
Yeah.
Or slide 5. We can start with that.
Yeah.
So this is the one that Mayuk generated, I think, in when he was working on this. And I think it's probably one of the notebooks.
Yeah, Mayuk. Yeah, so this is not actually from Mayuk's notebook. This is a very similar graph. Like Mayuk had a few demos that had graphs similar to this. So I had refactored some code to create a plot that is similar to his. But these are like on a different. These are on a different neural network. That is the ResNet 50, which is a different architecture and it has different hyperparameters. So yeah.
Okay, so then this just shows like the different subpopulations of cells. So like this A is the AB lineage, the E is the E sublineage. I mean, they're just the sublineages of cells. And then the predicted is the dotted line. So you're doing like a predicted versus observed. So you're observing it and then you're predicting using the model. You're predicting it, right?
Yeah. Okay. So what I wanted to highlight is the data set. Like the length of the data set is actually having an effect on the overall results of the model. That's what I wanted to portray.
Yeah, I see that. Then it's tighter, like in a large part of the
Yeah, and to the left, like if you look closely to the left, you'll find a couple of corrupt points. Like some points are going some points are all over the place. Like, so they are there because of some of the corrupt images that I couldn't really remove, which I'm working on right now, because the dataset itself, it contains like many corrupt images, especially towards the corners. So I tried to use OpenCV to remove them all, but I couldn't really do it. So I'm working on that right now.
Yeah, I remember when Mayuk was working on it last summer, there was a lot of, because it's not fully aligned, they're just collecting, microscopy data, they're just letting it run across different embryos, and they're doing the tracking, so it doesn't align perfectly, and you can't really do that anyways, it's just kind of like, you just take the observation.
Yeah, exactly. Because it's not aligned, so some photos are corrupt, so yeah. So do you want to look at some other slides?
Yeah, go up, I think one.
Okay.
All right, that one, yeah. So that's this is the blue dots are data points, red lines are model predictions. So it's basically like the blue lines follow the red lines. So it's kind of like so we're increasing the number of data points and we're capturing the model prediction. So the model prediction is like a distribution based, or maybe not distribution based, but it basically simulates the distribution and you get like this prediction, and then the blue lines match that eventually. I mean, but you might,
Yeah, you're close. Yeah,
you might expect, except that on the left you have 4 dots, so you really don't know that. At least with this, you know that it's closer. It's pretty well confirmed. So I think that's.
What I wanted to tell by this image is that more data always helps. It's better to have it in your disposal. So that's the goal.
Yeah, I think that's a good lesson. So I think that's good. And so go back to slide 2 for a minute.
Okay.
Yeah. So this is a nice sort of summary of what we have here with this data set. So this is collected on this group. They basically put a fluorescent marker in the embryos, and they tracked all the embryos across I don't know how many worms, like hundreds, maybe 1,000 or so, and they were able to get all these data, and they basically made all these videos available to people, and so it's in raw forms. It needs to be processed, but to have this like brought together as a annotated file is really good for future exploration, because they have like a version released where they've like put these dots, they've tracked them, put the numbers in a file, but the advantage of going back to the images is you can extract other types of information. So like with just the dot with the locations as points in a 3D space. Oh, hello. Oh, hi. The advantage to this is you can extract, maybe their extraction method wasn't complete, or maybe there's some features across regions of the embryo that we can recognize with this. So, I mean, this is a good upgrade. Now, Mayok isn't here. So we can't really ask him. Well, what you can do is just put this as a pull request, and then try to see if he can help you with some processing on this. I think having a processing power is always going to be an issue.
Yeah, exactly. I'll talk to him about this. Correct.
Yeah.
I'll do it soon.
So let's keep us posted on this. This is good work. And I like the idea that you've been able to expand the number of data points. I remember in the GSOC period, there are like constraints on what you can do in terms of time and getting up a proof of concept. So this is really nice that we can expand it out.
Okay, then, so you'll take over now, right?
Yeah.
Yeah.
Thanks for sharing.
Thank you.
Yeah, it's very good.
Yeah, absolutely. Thank you. Thanks a lot for having the time.
Yeah.
Oh, Bradley, I put a question in the chat.
Okay, let's see. So we have a question from Dick. Is there a measure of success? Can you simulate data and see how well it is recovered? So the measure of success, well, I think some of the curves he was doing with like making predictions of the data, like he's basically... Yeah.
I'm trying to be a little more explicit. In other words, if you pretended, even if the rules were wrong, that you simulated an embryo and got a whole bunch of cells with a differentiation tree, how far can you recover that tree from, in other words, if you put a blob at each point in space, each time you got a new cell, can you recover the coordinates of that blob? Or angles or whatever?
If we had a data set, we could process it further and put it in a normalized space.
Yeah, I mean, for example, you could make a simulation that said, okay, there's going to be a physical constraint, which is the size of the embryo.
Okay.
And within this, each cell is going to divide according to some program, and when it does so, it has to stay inside that boundary. Okay, and even though it won't simulate a real embryo, it will still simulate a branching structure within a confined space.
Right.
Okay? And can you recover at least the coordinates and perhaps the angles in such a simulation? Or does it fall apart? Does it fall apart after a certain number of cell divisions?
Well, I don't know. I think you would be able to recover it. I'm not really sure.
Yeah, well, okay. Well, I'm just suggesting an objective test where you can, where you know the data. The point is you know the data, and the question is, can you recover it?
Right. Yeah, I think I get it. Yeah.
Yeah, whereas you're dealing with data where you don't know the truth. You're trying to get at the truth.
Yeah, so you would create a simulation and then do it.
Yeah, produce the set of coordinates in space which are connected by their branching history. Or in other words, their differentiation tree.
Yeah.
Okay?
I think, that sounds good.
And that would give you some measure of how much success or how many generations of cells you can expect to go to before success becomes unpredictable.
Yeah. I'm just writing down notes here. Yeah, I think that's a good idea. Thank you.
Okay, sure.
Yeah, so that's good. Yeah, so we can do something like that as a test, because I know a lot of these algorithms, you get performance metrics for the data set itself.
Yeah.
Then mapping it to a developmental system is a little bit
different.
I
guess the rules would be the spatial constraints. Two cells should not overlap, and they should be outside the boundary of the embryo. Okay? Other than that, the angles could be arbitrary.
Right.
Okay?
Okay.
So it would be a very messy embryo, but but it would test the robustness of this technique.
Right. Yeah, it wouldn't be like there wouldn't be any sort of like biological structure to it necessarily.
No,
yeah,
no, don't expect any. You're not going to. All you're going to do is number the cells. You're not going to say what kind they are.
Right. Yeah.
Okay.
Yeah.
Okay, so it's a very crude simulation of an embryo, but it will test the robustness of the coordinate recovery.
Yeah.
Okay.
Okay. Krishna had a comment on, are you talking about the precision measurement? Oh, is this about the thing that we were just talking about, or about the graphs that we had?
Krishna, we're asking you, not me.
Yeah.
Simulating data and finding how well it recovers.
Yeah.
So I was talking that, so you were talking about finding the precision of the algorithm. Actually, I was not able to understand your question fully.
You mean my question?
Yeah.
Oh.
Okay, what I mean is this. Suppose that you knew exactly where each cell was at each time.
Okay, now I'm clear. So yeah, we can, yeah.
How accurately can you recover that information? And what I'm suggesting is making a simulation of an embryo. It would be a very crude simulation, but it would include precisely known coordinates and angles. And the question is, can you extract those from the simulation, even though the simulation is not a real embryo?
Okay, I think that more to accuracy, precision, or you can say specificity of the data would be a considerably better matrix for this, because I don't think that for such data we should not be focusing on getting 80% of accuracy, but we should be focusing more on what are the points that it's missing. So yeah, the thing you are telling can greatly improve the score. But our matrix, you can say error matrix, should be, I guess, specificity, or you can say precision. So we can call the precision and recall instead of the accuracy, and you can see where the simulation is going.
Yeah, but see, what I would expect is that your method will stop working after a certain number of generations of cells.
So you are suggesting an early stopping system. Yeah, that's possible. That we, generally don't train data to a certain limit. We, early stop it because there is a trade-off between the computation cost and the performance. If you can say spending $100 on computation is giving me 98% of precision, and then spending 100 again, it's giving me 98.1. And I'll prefer, early stopping. So yeah,
yes, I understand. However, computers are 10,000 times faster today than they were when I started using them. And but when you're my age, that will probably go up by a factor of at least 10,000 more. So, in general, I have no respect for computer time.
Yeah.
Okay. So arguing with computer time is arguing in the past, as far as I'm concerned. See, when I got started, there were physicists who would run a computer for 3 months to get an answer. And so, even though the computers were very slow then, one just ran them. That's all. I once proposed to a group that in order to discover breast cancer, we needed to run a computer for a whole week per person, and they just laughed at me. So, but I knew they were the fools.
Yeah.
If your things don't work, you people even have an excuse. And we don't have educate computation. We don't have any excuse right now.
Yeah, get a hold of a supercomputer if you can. There are some supercomputers that you can use for free online.
Yeah. I wish I was from your time. Every time a program didn't work, I can say that it's computational problem.
Hey, in my time, you had to carry a box of cards.
Yeah, my grandfather did his PhD from, I guess he's 87 now, and he also tells the same story.
Oh, yes.
And he always tells me that if these things would have been in his time, he would have done something very great.
Yeah, I remember. I had one mistake on one card, and the turnaround time was 24 hours.
Yeah. He told me how they used to, ship a 10 MB hard drive, and 5 or 6 people were used to carry it. And now we are having millions of, not millions, you can say that, thousands of megabytes in our pockets.
Yes, I agree.
Susan says, don't drop the cards.
Don't drop the card. Yes, I know a funny story of a student who waved to a friend of mine and dropped a whole box of cards. So, yes.
Yeah. So Krishna, did you have something to present today, or?
Yeah, I will. Okay. Actually, my mic on my laptop is not working, so I'm connected on 2 devices. I'm assessing the mic from my, so just give me 1 minute.
All right.
Brad, do you want me to present that winner gets killed presentation also? I guess that Dick would be interested in it.
Yeah, I can go over that too.
Okay, just 1 minute. How do I present? I think, Bradley, I think you should continue. It will take time for me to present. I guess I am not.
Okay. Well,
okay. I guess.
Yeah, I can see your screen, sure.
Okay. So, it is the same presentation that I presented in the previous week. So, is it the winner gets killed or the winner takes all? So, in an ecosystem, does the winner takes it all or the winner gets killed? So, here it is. Here it goes. Like, for example, if we are having 2 species that are competing with each other, so does the species that is winning due to some circumstances, like is it the ecosystem is more favorable, the other temperature climatic conditions are more favorable, so which helps in its survival, does it really wins or does the winner get killed? So here it is goes that, for example, we have 2 species and they are competing with each other, and if A's population is increasing, like we have we are in some place where cats and dogs are fighting, and cats can eat dogs and dogs can eat cats. So due to some reason, if the population of dogs is rising, then it will equate that the population of cats would decline. But this raises a concern that if dogs are getting greater in population, that would aid cats because their prey is also getting bigger. So if the 2 species are dependent on each other for their survival, so if the population of A gets increased, then the predators who prey on A will also getting increased because they have the probability of them finding a prey is even better now. That is Lokta-Volterra equation. So it's relationship between prey and the predator. So I get there was a fox which was used for fur, and that fox used to prey on a certain kind of rabbit. So when the population of that rabbit rises, the population of the rat decreases. And so was seen that when the population of fox was declining, to a certain extent the population of the rat increased, but then it's starting decreasing rapidly. So there was a relationship between the prey and the predator. So here it is, here you can see that if the various species are competing and they have their own predator, if one species flourishes too far above from the rest, then the predator also thrives and eats the winner. So if the winners are increasing, then the person who eats the winners are also increasing. So I think it should be forming an equilibrium. But in practice, models show that a combination of competition and predation eventually made all the species go extinct. So how this theory that just made perfect sense is not practically plausible. So then we have the concept it's not commonly proved. It was developed by Nigel Goldstein and Chai-Hyun, I know how it's pronounced. So okay, they developed the theory that when a species gets evolved, when a species is thriving, it's getting more prosperous. Then it's the species also modified itself and it's evolved. But so does its you can say its predator. For example, if the cats are becoming bigger, the population of cats is increasing, and they are also mutating, then there's the predator of a cat. So if there is not a co-evolution between them, they will get extinct, like the previous example we had. So if this was that when the co-evolution happens, it helps to sustain the whole equilibrium. Thank you. That's it.
That's good. So this is the Krishna's trying to come up with a proposal for the Evolution Conference, and this is the proposal. Last week we talked about maybe how you would simulate this to show maybe some results. I mean, there are a number of routes you could take. There are different software programs, agent-based models, or evolution simulations, or you could even do like a numeric simulation if you wanted. If you wanted to model like the Locke-Volterra equations, it's just like with two populations and have different scenarios.
And it is also known as paradox of the planktons. So.
Okay, I have a comment on this. There's a lot of literature on what's called a population crash, where basically a population increases, it wipes out its resources, then the whole population goes down generally to just a few individuals. This was first discovered using with deer that got isolated on a small island. They ate everything on the island, the population went up to thousands, and then it crashed to just a few individuals. Okay? The other thing on the mutations, that strikes me as being equivalent to what's called the Red Queen hypothesis. How many of you have read Alice in Wonderland? Okay, in Alice in Wonderland, the Red Queen shows Alice how she has to run as fast as possible just to stay where she is. Okay? And this has been turned into a hypothesis of how evolution goes on. That the predator and prey are both evolving, and they get better and better at running away from each other. Okay? So I put some references on both in the chat.
Okay.
On population and the Red Queen hypothesis.
Yeah. Yeah, they're over in the chat. Yeah, so there's some Sinervo and Svensson, Pagey, Hogaweg, Maynard Smith. Yeah, so there are some pretty good citations in there.
Yeah. Okay, so I think if you're going to work on this, you should know some of the background.
Yeah.
This
Yeah,
is not
sure.
based on a search. This is based on just what I happen to have in my bibliography.
Yeah. So yeah. Why don't you do some literature search and then think about how you might simulate this as a process. I think if you look through the literature, you'll find ideas for it, like how to set up the problem.
Yeah.
This is definitely a good way to go.
Yeah, I'm not aware of
Yeah.
simulations of both of these, but there may be some by now.
Yeah. I mean, they're not going to be all simulations. Maybe a couple of them have simulations in them, but you'll get a sense of the problem though by going through these.
Yeah.
Okay. Anyone else have any comments about this talk? We can chat about it later, like in Slack or whatever. And Krishna can, keep working on the slides. And so the deadline for that is March 1st. So it's a ways off, but not that far off. So if you just be mindful of deadlines. A month is not a long time, but you have to plan it out. So, okay. So that's good. Anyone else have anything else they wanted to present?
Bradley, could
Yeah.
you make some comments about measures and differences between bifurcating trees?
Would you like? Yeah, you can make comment. I mean, did you want me?
Well, no, I'm asking you.
Oh. I think I answered your email the other day about that.
Yeah.
Yeah.
The problem is we have very few differentiation trees, but we could generate some random ones and ask about similarity, stuff like that.
Right.
The question is, what is the similarity measure between two trees, and do we weight lower branches higher than more than higher branches, for example?
Okay.
Things like that.
Yeah. So yeah, we've done some. Well, we actually a couple years ago we did some work using the Hamming distance, which is an information theoretic distance. So the idea is you're comparing two binary trees, and you want to know if you have each node is identified with an ID, so you want to know if you have two trees and they're generated different, using maybe a different process or something, what the sort of match is between them. So if you have differences in the nodes, how have they rearranged themselves? And so it's actually, you can attack the problem in different ways. So about 4 years ago, came up with a method for doing this using a simple information theoretic metric. But there's no way really to say anything more than just the two trees are different at this amount of distance. So that's not really ideal, although it's a good start.
Is there any way of saying that they're similar up to a given point and then they diverge?
Yes, actually. So in phylogenetics, which is the study of evolutionary trees, so this is where you have common ancestors and then speciation events, and you have these trees that are generated over long periods of time, but they're inferred using common ancestors. Usually those trees are sort of the divisions between species are usually defined by traits that diverge. So it could be like a DNA sequence or a physical trait like an eye, shape of an eye, and you'll have what they'll call a meristic character or a character on each branch. And you can count the number of characters sometimes on a branch. So you might have a common ancestor. Let me see if I can illustrate this. I'm going to share my screen, because this is made for. So I'm going to use a tablet here to let me see if it's working today. Okay, good. So you have a binary tree, right? And erase this and start over. So you have a binary tree, and it has these, these divisions at different points. So let's say I have a binary tree like this. This is really not a very good drawing, but let's just work from here. So here's a node, here's a node, here's a node, and then the terminal node's up here. Okay, so our problem is we want to map, we have an ID for each node, so we might give it a number like, this is 1, 2, 3, 4, and then this is 5 through 8. And so the idea is that if we had another tree where, for example, these things flipped, so this were 3 and this were 4, then we'd want to know the distance between that tree. But if we do the single distance measure, we can't differentiate between the layers. We can only say that the tree is generally different. And in a phylogenetic tree, and this is using a method called cladistics, you get these trees where you get the same structure, but the differences are characterized by characters. So you usually put marks of characters on the branches like this. So you have a common ancestor here, right? And then there's a speciation event. So you have two different descendants, and the descendants are different by a certain amount. So this common ancestor changes in this daughter species by 2 characters, and this other daughter species by 3 characters. And so then you can measure this distance across the tree, and the same holds for these 2 branches here. So you can actually characterize, and these characters, they could be DNA sequences, which is rather objective, or you could use like some phenotypic character like eye shape, which is more subjective because you have to, figure out what the coding is for the shape and everything. But in any case, you get these differences in terms of the number of characters difference between the...
Bradley, simple question. Can these be mapped into what would be called colored trees?
What do you mean by that? Like,
in a colored tree, you assign colors to nodes or edges.
Okay. Yeah, I suppose. I mean,
There is a whole theory of colored trees also, or colored graphs.
Oh, right. Okay. I think I know what you mean now. Yeah, so we could do that. We could map that to a different problem domain. I haven't done this in a while. I was kind of fascinated by these at one time, like the idea of using, sort of the similarity between like cladistics, phylogenetic trees, and binary trees. And I never really got, much going on that, but that was something a long time ago. But yeah, you can take these types of trees and map them to other domains. So you could actually take this kind of measurement where you're looking at 2 trees. Now you have one tree here where you have these differences. Imagine if you had 2 trees where you had these differences. And this will often happen with, like if you have a gene tree versus a tree of phenotypic traits, where each source of data gives you a different answer. Or you might search through a search space for an optimal tree, and you have a number of candidates that are more or less the same optimality, but not really the same thing. So you get this, you're going to have differences in the number of characters on each branch. So you can actually compare those two. And the method they usually use in phylogenetics is consensus, which is just like an average. But there are other ways to do this, of course. You could characterize, you could, count the number of characters on a branch and like find a difference or find an average, or do it for each section of the tree, or map it to some other type of tree that's similar in structure. So yeah, there are a lot of ways to do this. I don't have an answer, but I think that would be something we could explore in further detail. I think in the future. I think there's a lot of potential application for it. I think Dick is looking primarily at we have the issue on complexity measures, and it's sort of a thing that we haven't really talked about. We talked about it on and off, but we haven't really done any heavy duty work on it. So we have some good references here: Meeks and Scott, Spanning Trees and the Complexity of Flood Filling Games. It's a theoretical computer science paper. And the complexity of networks to the set complexity of edge-colored graphs. So there are a number of problems, because binary trees are really just partitions of space and partitions of other types of things. So, I mean, we can like look at it that way too. We can look at it as like like KD trees are a partition of a space where you're dividing a space into halves recursively, and then the tree is a record of those subdivisions. So there are a lot of ways we could map like that problem to a problem like something similar to a phylogenetic tree, and then look at it that way. But yeah, I mean, there's a lot of ground to explore here. I don't really know. If anyone else has any comments on it.
Yeah.
I know Jesse had expressed some interest in that at one time, in the complexity measures.
It's quite off the topic, but I once read an article and it's described the human hierarchy with the help of phylogenetic trees, and that date back to when we and lobsters were having the same ancestors.
Right.
I can send the link. It's not exactly very biological. It's much more into psychology. But yeah, it's how one can use phylogenetic trees, and how our serotonin secretion and lobster serotonin secretion was linked due to some common ancestor, and how it explains human psychology and human hierarchy. I think if I can find the article, I'll send it.
Yeah, please do.
Okay, Bradley, the trees are also sometimes called partial orderings.
Okay, yeah.
I put a list of those in the chat.
Okay. We'll check the chat here. Okay, yeah. So yeah, there's some more references on partial orderings.
Yeah.
I'll make a record of those after the meeting here.
Okay.
And then Jesse says, yes, can't talk much right now, complexity. So Jesse's interested, but he can't talk by speech right now. So that's okay. We can follow up on it later. So yeah, thank you. We'll look into that further. I'm going to make a list of these references, and we'll probably talk about it next week more. Maybe this week we'll explore a little bit. But it'll be, that's this direction we'll think about in this direction. So actually, I can also send out the paper that we did in 2016 on this. It's not, like, again, it's just a small proof of concept, but it gives you an idea of what the problem is in terms of, like, looking at comparing trees. So let me go over some other things here. The last maybe 30 minutes. If you have to leave at the top of the hour, that's fine. I just wanted to go through some things here. So the first thing I wanted to point out for people, if you're interested, is that Louis Wolpert died this last week. He's a famous developmental biologist, and he had a lot of landmark publications in the literature. And I found this Web of Stories site where he talks about some of his stories here. Like where he was doing, I think he studied civil engineering at one time. Then he had a wild youth, and he switched to cell biology, and he did some work on the fluid membrane model. He's known for the positional information hypothesis, which involves like the position of cells in development. And so, it's a pretty key concept in embryogenesis. So if you want to read that, if you want to look over the Web of Stories, I put it in the chat. And you can also look up some of his papers. I mean, Louis Wolpert, search for that in your favorite library tool, and you'll find it. Another thing that's maybe coming up in the next couple weeks is that every year on this blog that I do a post for Darwin Day. It's Darwin Day is February 12th, and it's just a day where people usually, put together things, social media posts and things on Darwin. Allows people to think about, evolution and promote evolution and that. So this year's Darwin Day, we need a post for it. And I have some ideas, but if you're interested in collaborating on an idea for this, I'd be open to hearing about it. Last year I did a post on Sperry, Darwin, and the evolution of reference frames. So this goes back to the neuroscientist Roger Sperry, and he did these experiments with where they took a toad in development, and they took the eye and they cut the eye out and they rotated it 90 degrees or 180 degrees. So they took the eye, they cut around the orbit, and they moved it around like from where it was, and then they let the toad sort of behave. And the idea here is that the toad, when the eye is aligned as it is when it develops normally, it can target insects and catch them with its tongue. But when you do this surgery to the eye and you move the eye around, it actually flicks its tongue in the opposite direction from the fly. So it's suggesting that there's like this sort of automated response in the brain that's going on, but it's that because you shift that spatial reference frame, it's actually not registering the prey. It's doing exactly what it's supposed to be doing, but it's just not registering the prey in the right place. And so this is an interesting, it's called the chemosensory hypothesis, and kind of goes through, I kind of walk through that experiment and then how this might change over in evolution, talking a little bit about scaling laws and other things. It doesn't come up with a great insight. It's just, talking about these things that are related. And so then I have some references here. And that's basically what I would be looking for in a blog post. So if you have ideas about that, that's fine. Okay. Oh, so Dick spent a month with Wolpert in 1969. It's great. So
Yeah, long time ago.
yeah. And then Krishna can't open the page. Well, I don't know.
I got it. He sent me the right. He corrected the URL.
Oh, okay. Thank you.
The new URL works.
Okay. All right. And then so then that's okay. That's good. So if you're interested in this and doing a blog post, let me know. I'll be probably doing it. If no one has any ideas, it's fine. We might do something on like Louis Wolfert's work. It's possible. So then I didn't really get into this flash talk. This is a talk that I'm giving on behalf of the group next Tuesday. So maybe next Monday I'll have like a full dry run of this presentation. But this is it's a 5-minute flash talk. It's to this group. It's open science framework, because it's an organization that advocates open science. They run the Open Science Framework repository, and they have a conference on open source education. So this is a talk to open source educators, and this is supposed to be a 5-minute flash talk, which means it just has to have a lot of images, and you have to go through the slides quickly. But this is going to be on the DevoWorm platform. So I've got a bunch of slides here. I think I have enough slides. The question is how to organize this so that it's effective. So I've got a couple of, I have like the open source contributors that we've had on the platform. So we've had about 14 contributors over time. A lot of those were during Hacktoberfest, but some of them were just, like people come to the meetings, Ojwal, Krishna, Jesse, and, they've been contributing on some of the other parts. This just kind of goes through the genesis of this platform, some of the things that sort of, we're offering, machine learning models. We have this DevoLearn version 0.2.0, which is updated now, but, then this is a model of the I might modify this a bit, but this is a model of the DevoWorm program. I might actually update that to reflect maybe the whole platform. But this is actually a schematic of the whole platform. So this is actually now 0.2.1. And then this is this is the DevoWorm standalone software. And then these are the two other major things, although I might add in the data science tutorials as well. This is the species-specific models in the DevoZoo. So, there's this umbrella of things that we have for education and for research. And then, so I have a lot of visuals. I don't really have it organized yet. I'll have to do that this week. But I think it's coming along. I don't think, I think we have a pretty good, I should get some good feedback here, hopefully. And then that'll help us promote the organization as well as get some ideas for future development. So, I mean, it's something that, we'll see how that goes. So then finally, I have some other things here I wanted to finish up on. I have this really nice tectonic drift simulation. So this is a simulation that was just released. It's of the continents as they sort of formed as cratons about a billion or so years ago. And as the plates on the Earth's surface have shifted, these cratons have come together to form continents and then form the continents that we know today. So you can kind of see that things are starting to become recognizable as a map of the globe as we know it today. It looks like a bunch of ice cubes moving around on the surface of a lake or, a cup of water or a lake or something. You can see there's our world. So this starts at a billion years ago, and it's moving into the present. And you can see almost towards the end, you start to see the continents as they look today. And kind of in their relative positions. So that's from this paper here. Extending full plate tectonic models of deep time. Oh, actually, it's a little after the boring billion. So this is like the beginning of this is at the end of the boring billion. But I think it's interesting that, you have to consider that, the world is like the continents are, as it were, really weren't there in the same way that they are today. So, we kind of know from the simulation that there were a bunch of pieces of land scattered around the globe, and that it's much different than thinking about the world today, the biogeography of the world today, where you have continents and you have the oceans where they are. See, this is the way this might have some relevance to the boring billion. So they modeled this into deep time. You can read more in this Earth Science Reviews paper. It's brand new.
By the way, if you go really far back, there are things called microclimates.
Okay. Yeah.
Okay, or excuse me, microclimates.
Microcosms.
Yeah. There's one in the middle of the US in the south. It's about 800 kilometers away.
Yeah.
Anyway, the point is that people who think the Earth was covered with an ocean usually ignore these microclimates. So there was some terrestrial land even at very early stages.
Yeah.
So it'd be nice to see that simulation go backwards all the way to the beginning of it.
Yeah. Yeah. I don't know how much I don't know how much farther they can go back on this. I've seen simulations where they've gone back like a couple 100 million years, but this is the first one I've seen of a billion. So it's yeah. So that's interesting. Let me go back up to the main folder here. Okay. So the next thing here is, there's this, let's see, this paper here called Survival of the Systems. And this is a new paper in Trends in Ecology and Evolution. And I found this interesting. The abstract says, since Darwin, individuals and more recently genes have been the focus of evolutionary thinking. The idea that selection operates on non-reproducing higher-level systems, including ecosystems or societies, has been met with skepticism. But research emphasizing that natural selection can be based solely on differential persistence invites reconsideration of their evolution. Self-perpetuating feedback cycles involving biotic as well as abiotic components are critical to determining persistence. The evolution of autocatalytic networks of molecules is well studied, but the principles hold for any self-perpetuating system. So what they're getting into here is they're thinking about systems at a very broad level, and they're thinking about self-perpetuating feedback cycles, and they're thinking about autocatalytic networks of molecules. So they're kind of thinking about this sort of self-surviving aspect of a system, but at a very general level. So, they talk about ecosystems and societal examples, and persistence-based selection of feedbacks can help us understand how ecological and societal systems survive or fail in a changing world. So you have systems that are very general that they're talking about, and they're interested in what makes them persist over time. Because you think about, we think about maybe a system of feedbacks, and I know Jesse is interested in the cybernetic systems where you have these perpetuating feedbacks and other types of regulation. But, some of these systems have been in place for millions and millions of years. And if you're talking about ecosystems, like we just saw in the simulation of the different pieces of land and moving around, those systems can survive for a while and eventually they change. But what allows them to survive for long periods of time? What allows them, because they're not static, they change a lot internally, but why do they stay in a certain state? And so this is kind of what they're getting at here. So they say, recent theoretical progress highlights that natural selection can occur based solely on differential persistence of biological entities, without the need for conventional replication. This calls for reconsideration of how ecosystems and socio-ecological systems can evolve based on identifying systems-level properties that affect their persistence. And so feedback cycles have irreducible properties arising from the interactions of unrelated components and are critical to determining ecosystem and social system persistence. And so they get into self-perpetuating feedbacks, and cycles built from the byproducts of traits naturally selected at lower levels, avoid conflict. And so they go through this list. I mean, there's a lot of words here that I've gone through, but basically they look at a number of these things. It's a very theoretical paper. Feedback cycles as units of persistence-based selection. So they're looking at feedback loops. They're interested specifically at something called autocatalytic networks, which are networks of molecules that are sort of self-reproducing and self-regulating. If you know anything about so-called autopoietic systems, they're similar to this. I don't know if they actually cite that work in here, but they kind of go through types of self-perpetuating feedback. So when you're looking at specific systems, you can apply this theory. So one of the types of self-perpetuating feedback is resource acquisition and recycling. Another is, disturbance enhancement. So those are the two, I guess. And then feedbacks involving diversification and specialization. They get into that. This is a nice figure showing how self-promoting cycles support diversification. So there's this relationship between size and performance and diversification. So systems will grow and change but they're based on these rules that they're trying to sort of propose here. Finding stability is also interesting. So systems, with a lot of feedback, they're very dynamic, but they have to be stable or else they fall apart. And so they kind of give you they kind of go into that part here. And then the concluding remarks, the outstanding questions. So they have a number of questions here that are possible to come up with from this. So this is a good paper if you're interested in this topic. I thought it was interesting. I wanted to go over it a little bit and give people a taste of it. There's this other thing here, this paper on deep learning for reconstructing continuous processes. So this is a shorter paper. It's a Toward Data Science blog post. How deep learning is revolutionizing cell biology. So this is about kind of how deep learning is used in cell biology. As we've talked about it much in this group, there are a lot of open questions, and this kind of summarizes a lot of these questions. So one of the things they point to is distinguishing shapes versus distinguishing size. So they talk about this in the context of convolutional neural networks. And cell biology presents us with a very different problem from some of the benchmark data sets, and that is that the classes that we would like to categorize often vary not so much in shape but in size. And so that's something to keep in mind. And this gives you a lot of, I think, it kind of frames a lot of when we talk about deep learning in the type of data that we're dealing with in this group. I think it gives you a good primer on sorts of the issues, some of the issues you might think about. And I know Krishna is thinking about this general biological model. Is this something you might look into, Krishna, to get a sense of the problem space here? Let's see. Yeah, so CNN trained on these three classes must essentially learn to recognize circular objects on 3 different sizes. The remaining part of this article, they talk about cellular image classification, reconstructing continuous processes, and subcellular image classification. So they're looking at these circular objects at 3 different sizes. They're probably spatial scales in a biological sense, but in a data sense, they're different sizes. And so these are things that you're doing. You're either trying to reconstruct a process over many images, you're trying to classify cells in a single image or across images, and you're trying to maybe classify things within the cells. So you may be like mitochondria or other organelles that are in the cells, and classifying them not only by shape but by size, because things can be very similar in terms of shape but not necessarily in size, and that's sometimes the way you have to differentiate things. I would say that there's a lot of complexity, a lot more complexity than that. I think, like we've talked about, meta features in images, and those are things that exist across cells, across cell types, that maybe give us clues to like motion or other types of processes. So there's a lot more to it, I think, but I think this gives you a good handle on some of these issues. So they go through these reconstructing a continuous process, and these images are blurred when I printed out the PDF, but if you want to find out more, you can go to the actual article. So that's an interesting article. There's this other paper in Nature Machine Intelligence, A Biological Perspective on Evolutionary Computation. So this is by Risto Mikulunian. He's a big name in evolutionary algorithms, and Stephanie Forrest, who's a big name in complexity. I think she's involved with Santa Fe Institute and in that area of science. And they've published this new article on evolutionary computation from a biological perspective. So sometimes we talk about evolutionary computation in this group. So evolutionary computation is inspired by the mechanisms of biological evolution. With algorithmic improvements and increasing computing resources, evolutionary computation has discovered creative and innovative solutions to challenging practical problems. So evolutionary computation is where you have like a set of genes, they're computational genes, so they're usually strings of binary digits. And those binary digits then are used to encode information that would be encoded in a genome. And then you use these strings, you evolve them by replicating them and mutating them, and over time they give you solutions to problems. And so you apply this algorithm, you select on the best strings, you mutate them, you do all the things that evolution does, and you end up with these solutions. Now they tend to take a very long time to evolve solutions to converge on a solution. But that's one of the things but actually makes them very, that same property makes them very good for finding sort of creative solutions to problems. And so people have played around with these for at least 20 years now. And they're not in the mainstream, but you'll see them mentioned. So there are a number of things in biological evolution so they're not exactly biological evolution, but we want to always think about it in terms of biological evolution. And that's sort of the promise of evolutionary computation, is that it has a lot of the sort of characteristics of biological evolution. So in biological evolution, there are things like open-endedness, which is like where you don't really have a goal, you're just evolving things, and you end up at a certain place, but it's like you don't really know where that place is in advance. Major transitions in organizational structure. So if you have a transition, say from maybe like a single cell organism to a multi-cell organism, that's a major transition in organizational structure. Neutrality and genetic drift. So not everything in evolution is a product of natural selection. There's actually a lot that is the product of neutral processes, like the drift in certain genes to certain populations. And I'm not going to get into what those are, but you can look them up. But those are features that you don't necessarily get with a machine learning algorithm. You can also model multi-objectivity, which is to have two or more goals in the same algorithm. So if I wanted to optimize for maybe 3 or 4 different things at once, you could use the evolutionary computation to do that. Complex genotype to phenotype mappings, which are where you basically have this genome that I told you about, and you map it to some maybe set of shapes, which are the phenotype. And so you can do these type of mappings and evolve them and get a sense. Maybe sometimes a lot of people apply it to biological problems, but you could imagine maybe designing different types of car body or different program interfaces using the same method, where you just encode all the sort of encode templates into the genome and then express it in different ways, and you get a phenotype. So, I mean, and then coevolution, which is the evolution. I think Krishna talked about coevolution in his presentation, where you have 2 species evolving and affecting each other's fitness and their evolution. And so you can model all these things using evolutionary algorithms. Evolutionary computation exhibits many of these to some extent, but can be more can be achieved by scaling up with available computing and emulating biology more carefully. In particular, evolutionary computation diverges from biological evolution in 3 key respects. It is based on small populations and strong selection. It typically uses direct genotype to phenotype mappings, and it does not achieve major organizational transitions. These shortcomings suggest a roadmap for future evolutionary computation research and point to gaps in our understanding of how biology discovers major transitions. And advances can lead to, computer algorithms that approach the complexity and flexibility of biology and maybe serve as an executable model of biological processes. So this is a nice comparison of the 2 areas. You definitely have the algorithmic approach on the one hand, and then the life approach on the other hand. And this kind of lays out what evolutionary computation looks like. So I described it very roughly, but this is a good summary of it. And they talk about a comparison of biological and computational evolution across several characteristic dimensions. And so they talk about the different ways that you can simulate these different dimensions. There are actually programs that you can use. So HyperNeat, for example, can model genotype to phenotype mappings. Multi-objectivity, you can use a quality diversity algorithm. Neutrality and drift, you can use novelty search. Open-endedness, you can model with the Avita program, and so forth. And these are all programs that are available. They've been developed in computation and biology. So these different things are sort of their presence in evolutionary computation today. So major transitions are very minor in computation, but very transformative in biology. And so that's where we need to catch up. So this is an opportunity here. So open-endedness, we need to scale up open-endedness in algorithms. In major transitions, we need to develop that capability. Neutrality, we need to emulate biology more. Same thing with genotype to phenotype mappings. And coevolution, we need to scale up. And so this is a nice roadmap to how the opportunities in evolutionary computation. And so there are also some opportunities and challenges. So ECs have actually been around about 60 years, and they started with John Holland, who was a researcher at the University of Michigan, and he did a lot of the early work on ECs. But really, for the last 20 years, they've been a big deal. But if you want to go back and look at some of the literature, I recommend John Holland's book. He has, and then there are other people who have done a lot of groundbreaking work in these areas. But yeah, so if you're interested, there's some references in here. But also you have, there's a lot of new and exciting work going on. So if you're interested in this, I would follow up with this article, but also look in the literature a bit. And so I think Krishna's talk, I know he gave a talk on, or we gave a talk on the difference between biological neural networks and artificial neural networks at NeuroMatch. And so I think this is a nice follow-up to that, looking at the difference between like evolutionary algorithms and evolution. So, and you can see that there are a lot of maybe similar issues there in terms of the differences. Oh, you're muted. Okay, get a chat. Oh, carry on. Okay. I've been using screen print for later reference. So, I mean, yeah, actually, let me put the talks in a, let's see, I can do a link to the papers, and that's going to go away in a little bit, but you can make, and I can send out the link, and I can actually send out this entire list. We've had a lot of different references today on different things. So why don't I make a list after the meeting, and I can send it out. That way everyone has it, and they don't have to worry about whether I captured the screen or not. So yeah, that's it for today, I think. Any comments or questions on anything we covered? Are you there, Krishna?
Yeah.
Okay.
Can you?
Yeah, I can hear you.
Can you?
Yeah.
Okay. So I was saying that I guess in the previous meeting I told you about your presentation on form and factor, like the Euclidean cycle of life. So I think we can also consider it for for Darwin's Day or for the evolutionary conference. that how structures that are geometrically more stable are are organisms evolve into the structures that are geographically more stable, like how the bones forms structure and about the golden ratio in evolution.
Yeah. I'll have to. Well, I pulled it out a couple weeks ago. I was looking at it, so yeah, we can look and see if we can make like a abstract for it.
Yeah.
Any other comments or questions before we go? Okay. Well, thanks for attending. Next week we'll be back at the same time, and I don't know if we have any presentations. If we want to do, we'll keep tabs on Krishna's abstract. Maybe you can, develop it a little bit more. We can revisit it.
Yeah.
Okay. All right.
So, bye, Bradley. Bye, Dick. Bye, Susan.
Have a good week.
Bye. Yeah, have a good week.
Bye.
Bye, Mainak.